Hi, I'm Prof Eric So. I'm going to tell you about how our research can help to develop more effective treatment for severe form of blood disorder. Our lab is interested in understanding the molecular biology of normal and malignant blood stem cells. Stem cell is the most important cells that produce all the cell types in our body. In the blood system, hematopoietic stem cell sitting at the top of the differentiation hierarchy that give rise to all the white and the red blood cells. So it's not difficult to imagine that a lot of this blood cell defect can potentially be corrected by putting back, or in clinical terms, transplanting back these HSC hematopoietic stem cells into the patients. But unfortunately, bone marrow transplant is a high-risk procedure, largely because the patients may not be able to receive enough or fast enough and large quantity enough of hematopoietic stem cells to repopulate their populations, and they frequently will die because the blood cell wasn't repopulated properly. And so that's why one of the major interests in the lab is to understand the biology of normal hematopoietic stem cells. How can we manipulate the stem cells to allow them to produce more of the stem cell as well as their progenies, and particularly upon the bone marrow transplant? One of the prime and recent examples showing the potential benefit of our research to the patients is very well illustrated in our recent discovery and its publications in Blood Journal, which is the most prestigious and impactful journal in the field of hematology. And in this study, we have found out normal hematopoietic stem cells activity is regulated by the crosstalk between a group of protein called beta-catenin, HOX-A9, and PRMT1. And PRMT1 activity is absolutely essential to regulate the stem cell activities. Suppression of PRMT1 activity will result in stem cell depletions and excessive DNA damage of the cells. On the other hand, if we enhance the PRMT1 activity, it can rescue the phenotype and improve the stem cell repopulation ability and allow the cell to get into the replication to produce more stem cells, and particularly upon the bone marrow transplant situations. In order to make this work a successful example, actually we were able to pull together a group of very talented researchers with complementary expertise that's supported by the trust and also the college separately to work seamlessly to make this discovery. And indeed, without their support, this work would not be possible. So that is also another testimony for the long-term support or long-term visions of KHP to provide novel and effective personalized medicine to improve the patient care.